Sentaos y descubríos. Buenos días a todos. Se abre la sesión. Se va a proceder a la solemne investidura del profesor don Guy Smagghe como doctor honoris causa por la Universidad Politécnica de Madrid. Tiene la palabra la señora secretaria general de la universidad, profesora doña María Teresa González Aguado, para leer el acuerdo de su nombramiento. Por favor.
Yo, María Teresa González Aguado, secretaria general de la Universidad Politécnica de Madrid y en uso de las facultades que me confiere el artículo 68 de los Estatutos de la UPM y el nombramiento otorgado por resolución rectoral de 27 de abril de 2016 de la Universidad Politécnica de Madrid, certifico que en la sesión ordinaria del Consejo de Gobierno de esta universidad, celebrada el día 29 de noviembre de 2018 y a propuesta de la Escuela Técnica Superior de Ingeniería Agronómica, Alimentaria y de Biosistemas, una vez oídos los informes pertinentes y en reconocimiento a su amplia trayectoria y decisivas contribuciones en el campo de la ingeniería, se acordó por unanimidad nombrar doctor honoris causa por la Universidad Politécnica de Madrid al profesor Guy Smagghe. Y para que conste y surta los efectos oportunos donde proceda, expido y firmo la presente certificación en Madrid a 15 de noviembre de 2019.
Muchas gracias. Ilustrísimo señor director de la Escuela Técnica Superior de Ingeniería Agronómica, Alimentaria y de Biosistemas, por favor, invite a la profesora Elisa Viñuela y al doctorando que va a ser investido doctor honoris causa a acompañarnos en este solemne acto académico. Señoras y señores, levantaos y cubríos. Sentaos y descubríos. Tiene la palabra la profesora Viñuela, madrina del profesor Smagge, para pronunciar la laudatio del mismo, por favor.
Mr. Rector, Mr. President of the Social Council, Mrs. Vice-Rectors, Mr. Secretary General, Mr. University Ombudsman, Mrs. Directors of Senar and Departments, authorities, lecturers and students, Professor Smagge families, ladies and gentlemen, they are all. The Technical University of Madrid is very pleased to award Professor Guy Smagghe of Ghent University in Belgium the degree of Doctor Honoris Causa. This is the highest honor we are able to confer. Professor Smagge was born in Ghent, Belgium in 1968. He received a Master's Science in agronomy and crop protection in 1991, and 4 years later, a PhD in applied biological sciences, both with the highest distinction by the University of Ghent, the School of Bioscience Engineering. In 2012, he became full professor in the crop protection department of the same university. Today, we honor Professor Smagge for being internationally recognized for his excellent achievements in his career profession, developed in some aspect due to close cooperation with our university for 26 years. Professor Smagge is an extraordinary devoted researcher of agricultural entomology, a science sometimes considered with a minor recognition because the results of our investigations are sometimes published in index journals with low impact factors compared to other sciences. But it's a science with a major impact for society, because entomologists contribute to solving one of the key problems in agriculture, the control of pest organisms that threaten our crops and diminish the availability, the profitability, and quality. In the near future, it has been predicted that we will reach 10,000 million people in the Earth, and without agricultural entomologists, there will be a shortage in food supply. To raise awareness of the importance of plant health in addressing issues of global concern, the United Nations has proclaimed the year 2020 as the International Year of Plant Health. Plant health is protecting the world's plant resources from pests. Entomology is a vast and complex science with general, medical, veterinary, and agricultural branches. Professor Smagge's field of work within agricultural entomology had focused on several issues, general or applied, some of them clearly outstanding. For example, insect endocrinology, contributing to unravel the molting hormone role or the mode of action of some modern pesticides. Also, insecticide resistance, one of the main problems in our crops for the economic implication. He has contributed to know better some mechanisms of resistance. Also, he's working on the compatibility of natural enemies and pesticides. He's at present the convener of the International Organization for Biological Control Working Group, Pesticides and Beneficial Organisms. It's an international forum where researchers can exchange information on the joint use of these beneficial organisms and some pesticides. Concerning pollinators, he's internationally recognized for his key contribution to the role of bumblebees as vectors of microorganisms for the control of greenhouse pests. And finally, I want to mention RNA interference, a new topic at the time he started working. Some of his studies has focused on the control of some pests, coleopteran pests in Africa, and has been able to secure funding from the Foundation Bill and Melinda Gates. Since the 19th century, humankind has dismantled the natural world into smaller and smaller pieces, cells, molecules, atoms, electrons, and so on. But already in the same century, Haeckel, the famous naturalist philosopher who made popular in Germany the Charles Darwin theory on the origin of the species, were the first in stating the necessity of reconciling the fragmented world. Also, the famous botanist Muir, at the same time, were the first in realizing that all is important in nature, all is connected in life. Following in both scientist discoveries, Professor Smagge has managed to relate the different disciplines of the science of entomology in his works. He has devoted his career to advancing modern agriculture and sustainability by providing innovations in crop protection and pollination. He can be considered really as a polymath in the field of entomology due to his vast knowledge of many subjects around the world of insects. I want to highlight some of the skills of Professor Smagge because he has an extraordinary working capacity, an endless enthusiasm and curiosity, a great versatility, and an excellent sense of organization. Besides, he's also down to earth and has established ties with people from many countries, having at present a multidisciplinary and multicultural team. Overall, I would say that his accomplishments are credit to his positive attitude, being a highly motivated and inspired individual who enjoys working. Similar to the great naturalist Alexander von Humboldt from the 18th century, Professor Smagge also believes that with knowledge arrives the capability of thinking, and with this, the power, the capability to offer solutions to different matters through the observation, the study, the deduction. No doubt that Professor Smagge agrees with former USA President Thomas Jefferson's belief that tedium is the most dangerous venom on life. We first met at the University of Ghent in the year 1993 during my first sabbatical abroad. We soon established a fruitful cooperation between Ghent University and the crop protection unit of our university. This cooperation has evolved in many forms. For example, in the production of joint papers published in many top leading journals, in the form of co-supervising some PhD theses that has been recognized with prestigious awards, or with the training of the majority of my students in his lab for extensive periods of time. This cooperation has later expanded also to the Department of Biotechnology of my school, and some books have been jointly published in frontline publishers. Agricultural entomology is a complex world. Why? Because we deal with living multitrophic systems under realistic conditions. So we have the plant, we have the pests that threaten our plants and that sometimes transmit diseases, and we have some beneficial organisms that try to help us to control the pest. This makes our science distinctly different from other sciences because intrinsic features of living organisms inevitably lengthens the time to obtain a quality scientific publication. But in spite of this, Professor Smagge has been able to achieve astonishing academic results, well above the average, in a short space of time, 26 years. Really is truly impressive. His age index is twice as high as the top one Spanish researcher in entomology. His publications has been cited more than 20,000 times. More than half of his publications has been cited at least by 10 sources. Because of that, his I-10 index is also very high. He is author or co-author of more than 500 scientific papers published in international journals covering different fields. Also is author of many book chapters and has contributed to meetings and congresses with plenary lecturers, with oral or poster presentation. In addition, his creativity and innovation is confirmed as co-inventor of 8 patents. The last one from this year is a USA patent that describes a unique and innovative system, the Flying Doctor's Bumblebee Hives. It's a dissemination device. We use the bumblebees to deliver with precision some biopesticides, some microorganisms that control some pests in greenhouses. And this new technology is already being commercialized. But Professor Smagge is also actively engaged and invests a lot of time in the training of talented young students from anywhere in the world. To highlight the running project with China, 1,000 talent professors, aiming at training students and publishing in the top leading journals. And also, under his tutorship, more than 100 students have obtained the Master Science degree and 54 students the PhD degree. But still, he has time for the scientific community as editor of 14 leading journals. And also, he takes responsibility in the Belgian government, in the Belgian Superior Health Council with respect to plant protection products and biocides. Then, as expected, Professor Smagge has received an impressive number of international awards and recognitions for the excellence of his research. Very early, in 1998, was laureate from the Belgian Royal Academy of Sciences, Linguistics and Fine Arts. Some years later, he was elected Fellow of the Society in Vitro Biology, and some years later as well as Distinguished Scientist, just trying to highlight the importance of his contribution on the insect cell culturing. In 2012, he was selected Fellow of the American Association for the Advancement of Science. This is the largest scientific society in the world, with many Nobel Prize laureates, and publishes the top journal Science. This confirmed his major contribution to the field of entomology and agriculture, has been the second Belgian in being elected for this high honor after the Nobel Prize winning Professor Christian de Dief. He also has received 3 Doctor Honoris Causa degrees, and in 2017, he was elected Fellow of the Entomological Society of the America. It's the largest professional society of entomology in the world. Has been the first Belgian entomologist in receiving this highest honor. Also, in 2018, the Franke Foundation of his country distinguished him with the Franke Chair 2018. But to me, the most important achievement of Professor Smagge, the greatest possible recognition, is to have been included in the in the list of the highly cited researchers. We work in agricultural entomology. It's a small branch of science, but he has been included in this list produced by Clarivate Analytics. They also produce the list of the indexes journals. Clarivate distinguish or recognize the world-class researchers for their exceptional research performance based on the production of multiple highly cited papers. The list included many Nobel Prize-winning authors, and according to Clarivate, many others that really deserve being Nobel laureates. As a summary, from my point of view, Professor Smagge's success is largely accredited to many of the attributes that he possesses. He's a multidisciplinary researcher with a broad basic background, and this has allowed him to excel in diverse fields. Also has the ability to view problems from different angles, and this allows him to come up with solutions that otherwise won't be considered. But besides, he's also very collaborative and accessible, and cooperation has enriched our 2 universities for 26 years. Cooperation is always a mutualism activity that yields wonderful fruits. And as Alexander von Humboldt also said, all is interaction and reciprocity in life. Outside of work, Professor Smagge remains a dedicated husband to his wife Betty and to his 3 children, and all of them accompany him in this very marked date. And I do not want to finish without mentioning friendship. During all this year, we have been able to share wonderful moments all around the world. So, Professor Smagge, thank you very much for joining our university as Doctor Honoris Causa, for your tremendous contribution, support, and dedication to our profession. You have much to be proud of and many exciting opportunities ahead. And thank you, everyone, for your time and attention.
Muchas gracias a la profesora Viñuela. Se va a proceder a continuación a la solemne investidura como doctor honoris causa del profesor Guy Smagghe. Señoras y señores, levantaos y cubríos. profesor Guy Smagghe, el Consejo de Gobierno de la Universidad Politécnica de Madrid, en su convocatoria celebrada el día 29 de noviembre de 2018 y a propuesta de la Escuela Técnica Superior de Ingeniería Agronómica, Alimentaria y de Biosistemas, y en testimonio del reconocimiento de vuestros relevantes méritos, os ha nombrado doctor honoris causa de esta universidad. En virtud de la autoridad que me ha sido conferida, os entrego dicho título y os impongo los símbolos que acompañan al grado que ahora se os otorga. Asimismo, os impongo el birrete laureado, antiquísimo y venerado distintivo del magisterio. Llevadlo sobre vuestra cabeza como corona de vuestros estudios y merecimientos. Recibid el libro de la ciencia, conservadlo como símbolo de cuanto tenéis que aprender y enseñar, y que sea para vos recordatorio de que por grande que vuestro ingenio fuere, debéis rendir acatamiento y veneración a la doctrina de vuestros maestros. Recibid el anillo que la antigua universidad entregaba como símbolo de alianza con la misma y como emblema del privilegio de firmar y sellar los dictámenes y consultas de vuestra ciencia y profesión. Igualmente recibid los guantes blancos, símbolo de la pureza que deben conservar vuestras manos, signos también de la distinción de vuestra categoría y dignidad. Porque os habéis incorporado a esta universidad, recibid ahora en nombre del claustro de profesores el abrazo de fraternidad de los que se honran y congratulan de ser vuestros compañeros. Señoras y señores, sentaos y descubríos. Tiene la palabra el doctor honoris causa, profesor Guy Smagghe. Por favor.
Good morning. Buenos días a todos. Muchas gracias, dear Rector, leaders of University Politécnica of Madrid, officials, professors, colleagues, collaborators, students, many friends I see here. This is giving me a very good feeling, and also my dear family. I'm very happy, I'm very humble, I'm very proud on this to be here today. And I also want to start with this unique piece of art. I think it's also a proof of human intelligence and creativity. It was painted, as you see, by Anna Maria Sibelius Merian from 17th, 18th century. And it was not evident she was a woman. And she traveled to the Dutch Suriname. And she saw these wonderful small organisms, these small animals, insects, caterpillars, as you see, but also butterflies. And I think we can say when we look back in the history, before 1700s, she brought really color from the modern world, the new world, the Americas, back into Europe. And look to these colors, they are so wonderful. And they also show, and they inspired me also doing my research on the development of insects, how insects grow, and also how they do metamorphosis. So if you look to the title of her paintings, it's Metamorphosis of Insects, of course, from Suriname. And it's a wonderful developmental process, how you see that a caterpillar develops into a butterfly. And that, in fact, was also part of my PhD study. When you look to the metamorphosis, I think we can really say it's a wonder of nature, but it's also a wonder of life. It's happening in our small insects. It's not happening to us as human beings, but it's happening in these small insects, and most of them are quite old. So most of them, and even the most recent ones, most of them developed during the Jurassic period, which is 200 to 145 million years ago. So as human beings, I'm always very humble when I look to insects. Where do I do my research? Well, I was born in Belgium, in a small city in West Flanders at the North Sea beach. And I studied my graduation. I did my graduation in agronomy at the Ghent University. And I developed my research from physiology to ecology, and from lab work to work that is relevant to the society at global level. So I have my group at the Department of Plants and Crops. Formerly, we were named Crop Protection. And what inspires me a lot is that I do my research at different levels of organization. I'm happy to work at genome level, DNA level, RNA level, but also protein level. Cells and organisms, and of course my organisms of my study, or insects. Different insects, I work with bad insects, as we call them, caterpillars, beetles, like already potato beetle in potatoes, but also aphids, sucking insects. And next to the bad ones in agriculture, I also work with the good insects, like pollinators, bumblebees and wild bees. Also here in Spain, there is a lot of bumblebees. Yesterday I was walking with my family along the new park along the Rio River, and I think the diversity you provide there, the investment of the city, really stimulates biodiversity and also nature and pollinators. So I work with a lot of insects, different insects, and I also try to contribute at the level of the ecosystem. So in greenhouse, but also at ecosystem level, looking to pollination service, looking to the nature, but also looking to agriculture. Today, we are 15th of November. For me, this will be a day that I will never forget. It's a very honorable day and a day never to forget. And in Belgium, specifically, today is also King's Day. And you know, the royal family of Belgium has a very good friendship also with the royal family here in Spain. And that confirms again our good friendship already for 26 years here with the ETSIA of the Agricultural Engineers College. I prepared some slides. I prepared a presentation for you, not going too much in detail, but here and there I introduced some technical aspects. But there is one vision I want to bring, not only to you, but that I also use in my lab. And I want to support this with two examples. And the two examples serve, in fact, sustainability in our society and also biodiversity, because I think these are two important characteristics that we need in our society to invest in. So the first example that I give you is from the field of insect physiology and biotechnology. It's to support a more green agriculture with more sustainable pest control, less conventional pesticide residues, and a safer food production. We all know that our planet, the planet on itself is not increasing, but the number of people on the planet are increasing. We go to 9 billion of people, and we still have to produce food to provide the food in the supermarket or in small shops to make people to have a comfortable life. And the first technology I want to focus on is the technology of Rna Interference, or RNAi. And this technology really revolutionized not only the field of entomology, but also in medical field and also in the plant field. And it's because in 2006, the Nobel Prize in Physiology and Medicine was given to Dr. Fire and Dr. Mello for the discovery of RNAi, of RNA interference, which is called gene silencing by the use of double-stranded RNA in worms, in nematodes, not in insects, but in worms. Also in the plant field, there was a fantastic guy, Dr. Richard Jorgensen, who did post-translational gene silencing work in plants in Petunia. And he studied the regulation of the color in plants. And this, in fact, is the same principle. So you do not change the genome, but you destroy or you change in a specific way at RNA level what is happening. So to explain you a little bit, I go to the central dogma in molecular biology. So here you see a cell, here you see the nucleus with the DNA here, and this is then transcribed in messenger RNA. And what is happening now is that you destroy the messenger RNA in a very specific way, which is making that you can control the function of a gene. So you do a specific gene silencing. It looks a little bit technical, but it's a fantastic technology, a new technology, which allows to study gene functions without doing genetic modification. This is very important. We do not talk here about GMO. We talk here about regulation of RNA that the protein is not produced. So it's not a change of genome information. How does it work? And this is a little bit technical. You have a molecule, double-stranded RNA, and it's entering in the cell. You see here the outside of the cell, the cytoplasm, the inside of the cell with the cytoplasm. And there is a specific enzyme, which is called dicer. And this will cleave this double-stranded RNA in small molecules, which we call small interfering RNAs. And they have a very small length, which is 21 nucleotides long. That's a little bit technical, but you will understand that this is important because these small pieces will be recognized by another protein, which we called RNA-induced silencing complex. And this is by a protein which we call argonaute. This will be recognized and bound, and then this complex will search in the cell, in your cell, in my cell, in a plant cell, in a worm cell, in an insect cell, for the complementary part of this little part of 21 nucleotides. And this will happen in a very specific way. So it will search for it in the cell messenger RNA pool, and it will find there, and then there will be specific degradation. So what should you learn from this? There is no change of the DNA, so no genetic modification. One thing. Secondly, you search in a specific way to find messenger RNA inside the cell. And it's happening in plant cells, animal cells, and you can destroy this in a very specific way. So you guarantee the biosafety of this technology, which I think in terms of sustainability, this is very important. What can we now do with this technology with insects? I mean, this is, of course, looking to an application. Well, we can use it in protecting our crops against devastating insects that destroy the crops, like tomato tomato plants producing tomatoes or aubergine or corn. And we can target specifically beetles, but also some other insects. So I included in my presentation some examples to show you the potential of this technology. What you see here is the table. It's a very famous table. Every person in agronomy or in pest control has this table. It's a table from IRAC, which is not a country, but it's an international organization looking to Insecticide Resistance Action Committee. So it shows you all the different groups based on mode of action of insecticides, like pyrethroids, like organophosphates, like BT plants. And this table is in fact now outdated because with the new technology of RNAi, there should be a new group added to this table. And this is really a revolution. I cannot underline more. This is really a revolution that a new group should be added, and this is RNA-based biocontrol products. The big advantage is they are very efficient and they are very selective. They are very safe to the environment and also to human. So having said this, I give you some examples. And in my examples, I give you different ways how to deliver this technology for insect control in terms of sustainability. The first example is focusing on a beetle pest in corn. So it are beetles living on corn and they damage corn, and it's called one of the biggest problems. It's a 1 billion pest insect, and it's called Western corn rootworm. So the insect is eating the roots of the corn, and the corn falls down and there is no product, there is no produce. So people in companies like Monsanto company, they have developed a transgenic corn doing expression of that double-stranded RNA, and they are producing this. And you see here a corn plant, and these here, and this is a little bit technical, these are hairpins. And you see here a little structure here, and this is in fact the little structure of double-stranded RNA that is then eaten by these worms, by these insects, and this is then getting inside the insect, this little structure. And as I explained you, that little enzyme dicer is cleaving it in small pieces of 21 nucleotides, and this is then searching for a specific messenger RNA, which the target is here an essential gene, SNF7. But apart from the function of SNF7, what is happening is that this messenger RNA is cleaved down and the insect will die, let's say, within 3, 4, 5 days. So this is the mechanism where the product is delivered through an overexpression in the plant. So here we talk about a transgenic corn. And you can see here, this is a normal corn without protection. This is a corn that is overexpressing the double-stranded RNA, and it's giving a protection. So here you see a delivery through a GMO plant. The people of industry, they were very smart, and they combined overexpressing the double-stranded RNA together with an expression of BT to do a kind of a pyramid structure, also in the context of resistance management. So you can work on 2 different modes of action. So it's like you have 2 fishes for the price of 1. And this is now in the market since 2016. It was announced at the International Symposium of Entomology in Orlando, in Florida. And it's now 2 years in the market with great success. Of course, this success is in North of America, where the legacy on use of GMO crops is different than in Europe. And with my group, we invest already more than 10 years on the use of this technology, but as a non-GM crop, as a non-GM plant. So what do? We spray double-stranded RNA on the plants. And this is one of my postdocs, Olivier. And as a target organism, we use corroded potato beetle. So you know corroded potato beetle is coming from South America. Then it moved north to North America. And then it moved to Europe by a World War activity, we all know. And then here now it's also, in fact, it's a world pest, also invasive in China. So it's really a global problem, and it's one of the champions for resistance development. You can give Corrida potato beetle every insecticide that is on the market. It says, I like this, I'm not dying, I can resist. So there is really need to find new alternatives to control this insect. It's also a big problem. It's also a 1 billion pest organism, and it's resistant. So this is working. So we have very nice results where we can treat the larvae and also the adults to control them. So this is by spraying as a non-GM technology. I also collaborated with some people in US at USDA in Florida, Dr. Wayne Hunter and Dr. Eduardo, he's from Brazil. And we were focusing on psyllids. Now, I included this slide specifically for Spain here, because these psyllids, they are Asian citrus psyllids, Diaphorina citri, and they are vectoring a greening disease in citrus production. So like mandarins, like oranges. And it's also on focus here in Spain. I mean, you have a very important production of citrus. But I think also with the current problem in olive trees, where you have also the disease in olives, this also can be a technology to help controlling it. So we control this also by doing an impregnation of the product in a tree with an injection pistol. So we inject the product into the tree, into the sap stream of the tree, with a very good control. And the control goes for 2 to 3 months, which is really a very efficient technology. Also, non-GM plants, which allows to do agriculture and control in Europe. And another example, and this is my last example, where we also use the nature to deliver the product. And in the context also of biological control with viruses, we have been thinking to do the induction of gene silencing through a virus. So we engineered a virus to produce the double-stranded RNA, and then the virus could be distributed, and we did the work here on a fruit fly. The fruit fly, which is also a very aggressive and invasive species, Drosophila suzukii. Here you see it is in Europe, it is in Russia, China, and also whole North America. And it's also invading now South America and even in Africa. So this is how it looks. And it's puncturing the fruits on the tree. So apples, pears, cherries, every fruit that is on the tree is attacked by this insect. And the insect is putting eggs in the fruit. The eggs come out and the larvae eat the fruit from the inside. So we engineered the virus to produce this double-stranded RNA. And I show you here some results. This is results with cells, where you see here the control and here the virus. And you see there is an enormous effect for gene silencing. And the gene silencing goes together with a very high biological activity on the cells. But also on the insect itself, you see there is here a gene silencing. This is the control, and here a specific gene silencing of, let's say, 80% from 100 to 20%. And what is very interesting now is that when you look to the insect, to the flies, the adult flies, the survival of these insects in green, you see that they survive normally. But in the treatment, you see that after 6, 7 days, 8 days, all the flies that got infected with the virus, they are controlled, they are dead. And this is in fact the ambition also that we have to control these fruit flies. The advantage is, like I explained you before, it is very selective and safe for human and the environment. What is now important in this technology is that the product, the double-stranded RNA molecule that is carrying the activity, is stable, does not degrade. And we did some work on this with several of my students in the lab, and we see that this technology is not working in caterpillars. So we did some work, some basic fundamental work in bombyx, silk moth. You all know this nice insect producing the silk in the cocoon. which was in former times used in China, but also in Europe to make special clothing. And we were searching for the gene on this, and we were searching for a gene that is degrading this double-stranded RNA. So we were using double-stranded ribonuclease gene, and we were searching for it in the genome of Bombyx. It's a little bit technical. And we checked where this gene product was expressed, where it was present. So we did a lot of qPCR work, and we checked different tissues. And indeed, we found that in the caterpillars, it was highly expressed, highly present in the gut, in the stomach of the insect. So when the insect eats the product, there is a very rapid, very high degradation of the product. So the product is not active. And we also confirmed this with the protein, with the enzyme. So we took caterpillars, we dissected the gut, like a microsurgery of the gut. We took out the gut and did an in situ hybridization with the product. Oops. I hope this is not a sound from upstairs, that it's getting a little bit technical. So we did an incubation of this, and what you see here is the incubation in minutes, 10 minutes, 20 minutes, 30 minutes, and here you see the product. So this is the start of the incubation, 0 minutes, no degradation. 10 minutes, no degradation, but in 20 minutes, the product is completely degraded, which means the product cannot be active. The product is rapidly degraded by the enzyme present in the gut of the caterpillar. So this is making, there is a problem to use this technology in the control of caterpillars. But of course, we were searching for solutions. And we have a very good colleague in polymer chemistry at the Ghent University. He's a very good friend. We were searching for 2 years to find a solution for this problem of metabolic degradation. And we found a guanylated polymer. The structure is a little bit here. And we found that indeed this polymer is protecting this molecule even up to pH 11, which is extremely alkaline. I mean, you all are from different fields, but if you see a pH of 7.5, which is the normal pH as we have human beings in our body, a pH of 11 means more alkaline condition, more than 1,000 times more alkaline, because this is in log values. And this is extremely alkaline. And you see that the protection even is better in the higher pHs, in the more alkaline condition, which is very important, because the gut of a caterpillar is very alkaline. So with this system, and we published this last year, with this system we can find, or we have a solution to make this technology to be efficient in the control also of caterpillars, like we have many caterpillars in vegetable production also here in Spain. So what are my home take messages on this new technology? The RNAi is very useful in research, in fundamental research, because it allows to study the function of genes. Because you can do kind of an de-orphanization because of the high specificity of the genes. Today, we have already several products in the market based on RNAi technology, and more will come. I think also more will come to solve problems, different problems in insect pest control, but also in disease control, phytopathology, control of viruses, and even control of weeds. What is also very important, what is the cost to produce this? So there are different patents now, and also different production systems that are very cost effective. I remember the first experiments we did in my lab 15 years ago, and I paid a few thousand euros to produce a few milligrams of product. Now the cost to produce a gram is something like $10. So in a period of 15 years, the production cost has really decreased, which is making it possible to use this technology in practice. And then the last, on the stability, the metabolic stability and the uptake, these are very important factors, and I think we have contributed with my lab really to find a solution there. But of course, there is still knowledge that we still need to collect, and knowledge that is needed about good target genes. We developed a lot of things on bioinformatic pipeline, also in my lab, but also in other labs worldwide. Also on this uptake and this stability against degradation. So we use different polymers, biomolecules also from nature, and I think also interaction from physics and other disciplines are very important here. Also the safety, there is a lot of work to be done on safety for human and environment, non-target species. We use a lot of bioinformatics, big databases, also tiered approach experiments with different insects. And what does this serve? Well, it serves to use this new technology in agriculture. And of course, the data that we collect is science-based. And I think this is very important because nowadays, when you make a statement, it has to be science-based to provide trust with the stakeholders in agriculture as farmers and consumers. I think this is really something also as a mission and as the job, as a responsibility of the university, professors, postdocs, pre-docs, but also technical people at university to contribute with science-based research to make decisions and in fact to give trust to the stakeholders. In my field, which is agriculture, but I think it counts for every field at university. And I think the university should play a big role in this. On my contribution on the biosafety, I'm happy to show here the report we did for EFSA, the European Food and Safety Authority. We did this with a very large consortium with 4 different institutes, and it's available at the website of EFSA. And it's confirming again the biosafety of this technology in insect control. We also developed a cost action, and this is really to stimulate interaction within Europe as a network for senior people, for professors to visit another lab to learn a new technology, but also for young people to learn in another lab another technology or to bring new insights. So this is also open, and we always have quite a lot of people from the Mediterranean traveling to the north or to the south to learn new things, which I really think is very important. So this was my first example on sustainability in agriculture. I prepared a second example, and I hope you're still enthusiastic. And this second example is focusing on biodiversity, and I use here pollinators and pollination services. Because without pollinators, there is no food. There are no flowers. And it's a research domain in ecology for sustainable agriculture, but also for nature conservation. Like I started my talk on the green park along the Rio River here in Madrid. Why do we need pollinators? And specifically, I want to underline wild pollinators. Honeybees are one pollinator, but there are many more pollinators in our ecology, in our system, in our nature. What do? Well, they have an ecological benefit. They do pollination and they provide the guarantee for plant diversity. We call this weeds. But these weeds also produce flowers, and pollinators, they have a specific relationship to do the pollination. Also, there is an economic benefit in agriculture. I think tomatoes, coffee, and many fruits that we eat in the morning for breakfast, papayas, passion fruit, and even mandarins, they are all pollinated. But what is happening? There is a decline of pollinators, although they have an important service. There is an estimation by IPBES, which is founded by United Nations, that the pollination service, which we get for free from the wild bees, we don't have to pay for that. They fly around and they do the service. But if you calculate this, you have about a value, and this is really a lot of money, 153 billion of euros per year, that this is the value of pollination by wild bees. This is an enormous amount of money. And it's indeed worldwide, of course, and it's indeed to guarantee food production, to feed 9 billion of people on this planet. Because we have a limited land surface and water resources. And I think this is also something we have to give as information to our politicians. But bees, bumblebees, wild bees, there is a concern. I mean, everybody knows here in the audience, there is a concern. There is a red list, for instance, of bees in Europe. And when you look to the regression status of European bumblebees, 46% is under regression, which is nearly half. There is a pressure on them to disappear. And when you look to the number that is really under threat to disappear, I always compare this when you see a promotion of the World Wildlife Fund, WWF. They always use elephants as the example of animals that are under threat. Of course, it's very appealing. But also, 24% of the European bumblebee species are threatened with extinction. So this is also a big concern. And I think also university should make an action there. What are factors of decline? Well, our environment is changing. The habitat is fragmented. And of course, food. No food, no life. I think this is simple. When we have no breakfast, we feel hungry. When we have no lunch, we feel more hungry. And if you do this in a repetitive way, you don't survive. So no food, no life. This is very simple. Climate change. I'm not talking here about what is the reason of climate change, but there is really climate change. You see change of temperature. You see change of frequency, of hurricanes, of higher and lower temperatures, of rain. I think also here in Spain, you had a stronger frequency and of heavy rain and very dry temperatures. So also this is affecting the botany, the flowers, the food, and also the life of the wild bees. Intensification of agriculture. Of course, this has impact. Of course, this has impact. The monocultures, large fields, also pesticide use. And this is the next factor, pesticide use. I think everybody knows from the 60s the book of Rachel Carson on DDT. This is already a long time ago. It's still used as one of the key examples. But we also know the rules or the mandate that was given to the European Parliament for the ban of neonicotinoids with the specific example of imidacloprid. Of course, pesticides, specifically insecticides, have an effect because they were designed to control insects. The trick is to use biosafe insecticides like RNAi technology that is not damaging wild bees. And what is also very important, and we also investigate this in my lab in Ghent, is that with a driver on genetic level, so there is also low genetic diversity by inbreeding. And of course, this is together with the habitat loss. If you have a population of wild bees and the habitat is getting smaller, you have inbreeding. And the inbreeding is leading to a loss of genetic diversity. I call this genetic pauperization. And this is also a factor that is playing that after too much inbreeding, you lose your population or the population is losing the potential for adaptation. And at some generations, it's lost. And every year there is one generation. So after 10 years, it means 10 generations, the population can just disappear because of a genetic collapse. So these are all multifactorial and they work together. They are all local factors, so it means they are depending on what's happening at the local level. In Belgium, in Ghent, in Spain, south of Spain, Almeria, very large production of vegetables, or even here in Madrid. What can a society do? What can a city do, like a big city here of Madrid, to stimulate indeed or to counteract against habitat loss and stimulate with flowers, specific flowers? There is still one factor that I did not put here in the list, but what is the impact of pathogens, diseases? If we get a pathogen, if we get infected with a virus, for instance, influenza, we are feeling sick. And if it's very bad, like you had the Spanish fever after the Second World War, many people died. So also the bumblebees and the wild bees, they can suffer from pathogens. And pathogens can be viruses, can be bacteria, can be protozoa, fungi, or mites. And for this work, I want to give you an example, and it is an example where we went into the footsteps of Alexander from Humboldt. Of course, it was his work was in 1800s when he went to South America, where half of South America was still not described. But we also went to South America, and we went specifically to the region of Patagonia in Argentina. And we did work on this fantastic animal. It's the golden giant bumblebee, Bombus del Bombi. It's a specialist, and it's pollinating a native herb, Amancai, Astromeria. And it is the largest bumblebee on the planet, so it is about between 4 and 5 centimeters long. It's an enormous, beautiful animal, flying around, but it is disappearing. There is an enormous threat on disappearance of this fantastic bumblebee. And we investigated if there was a spillover of pathogens, and we were searching for a pathogen, Apicystis bombi. And this work was published in different journals. We also collaborated with people in Argentina. I like to do collaborations because I believe really in science without borders. And it was also brought into Science, where it got a lot of attention. And the title of the paper in Science was The Plague of the Bumblebee. And how does it come? Well, it comes that a lot of bumblebees in the 80s and 90s, the 1980s and the 1990s, were shipped to South America from Europe to do the pollination service in greenhouses for tomatoes. Now, at that time, in the 1980s, 1990s, there were no veterinary rules, nor there were diagnostic tools to do detection of diseases, of pathogens. So people were producing and they were shipping, and the bumblebees in South America were doing the job for pollination, for instance, of tomatoes in greenhouses. Now, from time to time, we know greenhouses are not hermetically closed. From time to time, there was a bumblebee escaping and flying out of the greenhouse in the nature. And then after some time, these bumblebees, they reproduce and they spread more. So when we look to the native bumblebees, Bombus del Bombi, they were here in Patagonia. So this is part of South America here. You see here Chile, and this is Argentina. And we were searching here in this area where you have the Andes, and the bumblebees was going through the valleys of the Andes from Chile into Patagonia. The non-native species of bees were honeybees, so they arrived in the 50s, 60s, and 70s. In the 80s, we had the bumblebee species Bombus ruderatus, and in the 90s, Bombus terrestris got also established in the region. Now we know this, and we collected samples, old samples, and current samples, modern samples, and we did some genetic analysis. So this is also where I think that the modern technologies in genetics, molecular biology, can help to solve old problems, can help to find a solution to problems that we could not investigate and we could not understand without the modern technologies. So to make a long story short, on this work we have been working about 3 years. So we collected bumblebees and we collected parasites from Argentina in this area of Patagonia. We also collected bumblebees and diseases from North of America because we wanted to know if the pathogen and if the bumblebee with the pathogen maybe was coming from the north, going from North America, Central America to South America, whether there was a migration north to south, which is possible because we know that some pathogens, they can fly from North of America to South America. Think about these butterflies, monarch butterflies, that fly from Canada here to Mexico and then to overwinter, like you also have Canadian people. They escape the winter time to stay here in the Gulf of the Caribbean Gulf at Mexico, and then when the winter is over, they go back north. So we know that insects, they can go from north to south of America. So that was our first hypothesis. Is there a spillover from pathogen from north to south? Or we collected also bumblebees and pathogens from different countries in Europe, from Belgium, UK, Ireland, and also from Israel here and in Europe, to investigate if indeed there was a spillover over the Atlantic to South America. So this was our material and our two hypotheses. And we investigated by doing genotyping. So it's a relatively modern technology where we do genotyping. And what you see here in the red are the lineages, the samples of Central America, of Mexico. In blue, you see the samples of Europe. And in green, you see the samples of Argentina. And what is clear is you see a mix of the green and the blue, which is indicating that the parasites that we found back in Argentina, in fact, have a common ancestor, have common material, genetic material with European samples. So this made us to conclude that based on our genetic studies, we could unravel that there was a shared origin of the pathogen in the Bombus del Bombi between Argentina and Europe. So it allows that to say that we try to understand the route of transmission in the global pollinator business in the 90s, that there was indeed transmission between Europe and South America. So in other words, the transmission was not from North America to South America, but the transmission of the pathogen was from Europe to South America. It's somewhat similar that the native people in the United States, in North of America, the Indians, more than half of them passed away, died because of the infection of measles that came together with the early pioneers coming from Europe. So this is exactly the same route of transmission with bumblebees from Europe to South America. And this is, of course, through the modern technologies that we could unravel the route of transmission of these pathogens. So what did we learn in the last 15 years of the work that we are doing with pollinators and their pollination service? So wild bees, they are important. They represent the value of about 153 billion euros for the pollination service. They do for free. Luckily, we don't have to pay for that. Otherwise, they will increase our taxes maybe by 2 or 3-fold more. This is a service to nature and to agriculture in our agroecological system, and this is confirmed by IPBES from United Nations. What do we also learn? Within Europe, we have a red list of bumblebees, and 24% is threatened by extinction. That is really an alarm, really an alarm. There is a loss of biodiversity and a decline of species, and there is different factors, different drivers, and they work together. It is not one driver, it is a multifactorial complex of drivers. What is also important is biodiversity is more than just a pretty face. It's not by having some flowers in a garden that you solve the problem. You need to have, again, a science-based design of the flowers. And with this, I mean that some flowers are good for some bumblebees or wild bees, some other flowers are good for other ones. So there is really a design needed, some science is needed here. What is also important that if you have bees in the city, they make people happy. So also bees, wild bees, they have a sociological role to play. And I think we should not forget this. Otherwise, we really end up in a very sterile urbanization environment. In our globalized economy, there is transport of pathogens and diseases, also exotic and invasive species. We all know this. In the 80s, the 90s, the technology was not there. But I think today I can say that today the veterinary rules exist and also molecular biology is contributing for a better diagnostics. So this is really helping. The science really has been helping also to prevent the transmission of diseases and invasive and exotic species. So diagnostics is very important here. And this came really from science. What about pollinators? Well, pollinators we can also use, and in the talk of Professor Vinuela, you also saw I developed something also as a precision delivery that pollinators can bring products to the flower to solve a problem in flowers. We call this flying doctors. It's now on the market. And it's to solve problems in the flower, to control diseases, and this is also reducing pesticide residues. There is no spraying cost for the farmer because the bumblebees, they fly to the flowers. You don't have to pay them. There is less fossil fuel burning because you don't have to ride with a tractor. There is less carbon dioxide emission because the bumblebees, they fly around. You don't need a tractor or a spraying device. And there is less water use, which I think is also important because when you do a spraying of a product, easily you use 1,000 liters per hectare. So no water use is necessary here. What is still needed? Some knowledge. And still we can contribute from the university there. In the different causes of decline and the interactions, like I explained, habitat, climate change, pesticide, pathogens, and the interactions are important. We still do not know everything yet. Also, how to measure the health of bees. When we go to the hospital, most cases, they take some blood and they do some testing to see if you have a disease, influenza, or if you have cancer, or if you have another disease. Well, also in this world, with our tiny insects, we are doing this. And with one of my PhD students, we realized something which is really contributing. It's based on metabolomics and machine learning. And we developed a diagnostic toolbox where we can measure the health based on metabolism, which is very important. It's like diagnostics for diseases with human. So this is recently out. It's very fresh. It's from August 2019. It's the first report in this field in insect research. And like I explained you, flowers are important to provide food to our wild bees, and the flowers get back the pollination service. But not every flower is appropriate. So you need to find the balance, science-based, between specialist and general pollinators to have a successful conservation. So some flowers, combination of plants are important to support the biodiversity of insects, of wild bees here specifically. And then the last, we also work on models, computer models, to predict and to increase the pollination service of wild bees. This is very important. We work also together with growers, where we tried to provide guidelines based on modeling to increase pollination service. On pollination service, we also started a cost action. It's called SuperBee, and it's looking to sustainable pollination in Europe. Also there, a lot of people from Spain are contributing. So these were my two cases that I wanted to share with you from the science we do in my lab over the last 15 years, with some moments and some take-home messages. But overall, I believe that with university, we contribute in education of young people and more senior people. And also we develop technologies. We use technologies to investigate biological questions, but also we develop technologies. I like very much interdisciplinary research and international research. Science without borders is really something I want to confirm that this is crucial, of crucial importance. What is also necessary? Fascination. You need young people and senior people with a fascination, with dreams. And of course, a very magic word, serendipity. You can plan everything, but from time to time something happens. Even when an experiment fails, you learn something. Even experiments without a result can be of interest. International, I think this is symbolized in this globe. Fascination is symbolized in this famous book from Don Quixote, from Cervantes. And then the serendipity. Every time I come here, I like to go to El Prado, and I like this painting, El Bosco. I like this painting. It's, of course, heaven and hell, fascination of life, but it's always, you find something new. I've seen this now, I don't know how many times, but every time you find something new that you did not expect. This is really the serendipity. And to quote also a famous Nobel Prize winner in chemistry, it was the serendipity, it was the luck, and some major failures that led me to the key, aha, the eureka moment. And I think this is really true. When I look back, I graduated in 91, so I'm at university 28 years, and this is really true. You can plan a lot of things, you need to train people, but never forget serendipity. It's so important to find also new things. With this, I also thank my family for working together, for their love. I mean, you also need this, to have somebody at home. This is a picture of my group. It's very international. You see a lot of colors, a lot of nationalities. It's about 50-50 national, international people, and also about 50-50 in gender, men and women. Different master students, PhD students, some technical support people, of course, also to support the administration, and 7 postdocs. This allows me to have a nice job, a job where I hope, I think everybody knows this statue from Rodin, the thinker, think, believe in something, dream, and dare to do. And what is very important, friendship. And with this day, I really want to underline this. The collaboration here with the university, the Polytechnic University of Madrid, the UPM, is already 26 years, living very active, and I hope many years more. And this is my last slide. I hope I didn't bore you. I hope I could transmit something interesting that you will remember. And this was a picture in 26 in Orlando at the International Conference of Entomology. And this is surrounded by the animals that give our job, that give our opportunity to work, also to be here today. And of course, you see here Elisa, you see here Josep, who was also one of the people in the early times in the lab. And this is, of course, me, surrounded by our insects that give us a fantastic job. With this, I thank you very much, and I really carry the friendship with Madrid and university forever. Thank you.
Secretaria General de la Universidad Politécnica de Madrid, presidente del Consejo Social, vicerrectores, responsable de profesorado y gerente, vicerrectores adjuntos, directores y decano de escuelas y facultad, directores de centros e institutos de investigación, profesor Smagge, profesora Viñuela, madrina del doctor Honoris Causa, familiares del profesor Smagge, profesorado, personal de administración y servicios, estudiantes y estimados todos. Celebramos hoy la investidura como doctor honoris causa del profesor Guy Smagghe a propuesta de la Escuela Técnica Superior de Ingeniería Agronómica, Alimentaria y de Biosistemas, y actuando nuestra profesora Elisa Viñuela Sandoval como madrina. Querido profesor Smagge, querido compañero, como digo siempre en estas ocasiones, bienvenido al claustro de la Universidad Politécnica de Madrid. Permítanme ustedes ahora cambiar unos instantes a la lengua inglesa. Dear Professor Smagge, thank you very much for having accepted this recognition award from Universidad Politécnica de Madrid as Doctor Honoris Causa. It's a pleasure and a honor for us to incorporate you as member of our university. Now, as well yours. Your portrait will be included into the gallery of Doctor Honoris Causa as model of excellence for our faculty. Now, let me switch again into Spanish. Querido Guy, tu nombramiento e investidura hoy como doctor honoris causa, máxima distinción académica que otorga nuestra universidad, es un honor especial para nosotros, dado que además, desde hace ya más de 26 años, colaboras de forma regular con nuestra universidad. Querida Elisa, muchas gracias por haber propuesto la candidatura del profesor Smagge como doctor honoris causa. Tal como nos has descrito en tu discurso, el currículum y la trayectoria vital del profesor Smagge es ejemplo para nuestros jóvenes investigadores e investigadoras que inician su carrera científica en un campo que, como también has mencionado en tu discurso, querida Elisa, puede aparentar ser pequeño, pero que, como casi todas las cosas en biología, tiene una relevancia fundamental para la supervivencia de nuestro mundo tal como lo conocemos. En las ciencias de la vida, agentes minúsculos, microscópicos, tienen una importancia vital. Reconocemos hoy a un ingeniero de las ciencias de la vida, un ingeniero especialista en agronomía y protección de cultivos, doctor en ciencias biológicas aplicadas, que ha desarrollado su actividad investigadora en Bélgica en los más reputados centros de investigación y ha sido capaz de extenderla a través de sus numerosas colaboraciones docentes y científicas a todo el mundo. Reconocemos hoy no solo a un impecable científico, sino también a un profesor que ha sabido extender de forma entusiasta su magisterio, y reconocemos a un profesional en un campo donde la transferencia de los resultados de investigación a producción se produce de forma casi inmediata por necesidad. Reconocemos asimismo hoy a alguien que cumple con excelencia las 3 misiones fundamentales y con el elemento diferencial que crea una universidad, que es la generación de nuevo conocimiento para que a través de su transferencia a la sociedad y a los nuevos estudiantes que formamos en nuestras aulas, contribuya al avance de esa misma sociedad. Querido profesor Smagge, querido Guy, muchas gracias por haber aceptado la propuesta para incorporarte a nuestro claustro. Querida Elisa, gracias por proponer el nombramiento del profesor Smagge para su investidura como doctor honoris causa por la Universidad Politécnica de Madrid. Gracias por vuestro trabajo en ese campo de la ciencia que, como he comentado antes, has afirmado cuenta con reconocimiento menor, opinión que, permíteme decírtelo, respecto de la palabra menor discrepo y no comparto. No podemos medir la importancia de un logro científico o de un trabajo exclusivamente por el índice de impacto de la revista en la que aparece publicado. No podemos, cuando alguien nos señala la luna, quedarnos mirando el dedo que la apunta. Dado que de ejemplos ha ido hoy la conferencia del profesor Smagge, esto no es más que un ejemplo. Y es la luna lo importante, no el dedo que la señala. Vivimos tiempos en los que parece que se valora más el indicador del logro que el logro en sí mismo. Tiempos en los que se cuestiona el papel de las editoriales científicas y el oligopolio existente en alguna de sus publicaciones. Tiempos en los que se empiezan a valorar nuevos mecanismos de colaboración en la comunidad científica y en los que la propia Comisión Europea nos anima a implicar a la ciudadanía en nuestras investigaciones a través de los SWAPs. En tiempos en los que tenemos que tener en mente los Objetivos de Desarrollo Sostenible de Naciones Unidas, tenemos que ser conscientes de que este planeta solo podemos mantenerlo colaborando y compartiendo el conocimiento entre todos. No podemos valorar la importancia de las actividades que realizamos exclusivamente por el número de investigadores o científicos involucrados. Y en el caso de la entomología y el control de plagas, soy especialmente consciente porque hace muy pocos días, además, albergamos en nuestra Escuela Técnica Superior de Ingeniería Agronómica, Alimentaria y de Biosistemas, albergamos el Congreso Nacional de vuestra especialidad, donde pude comprobar que constituís una comunidad entusiasta y comprometida con un trabajo que es, en realidad, por todos y para todos. Además, investimos hoy como doctor honoris causa a alguien vinculado a uno de los campos de la ingeniería menos conocido por nuestros estudiantes de bachillerato. Una ingeniería que, como pone en el lema de la escuela Sine Agricultura Nil, sin agricultura nada, y que, como habéis destacado en vuestros discursos, tiene un impacto fundamental sobre la supervivencia de nuestra sociedad y de nuestro planeta. Una ingeniería que es fundamental para el cumplimiento de esa Agenda 2030 que hemos firmado todos. Una ingeniería que nos debe permitir alimentar a la población mundial. Una ingeniería básica para eliminar esa lacra que es el hambre que soporta un porcentaje muy amplio de la población mundial y que, hablando de personas, no podemos escudarnos en los números. Cuando la FAO en el año 2016 daba el dato de que 815 millones de personas pasan hambre en el mundo, no podemos olvidar que cada una de ellas tiene un nombre, tiene cara y ojos. Son unos padres desconsolados porque su hijo, su hija llora y está en riesgo de desnutrición. Y cuando las estadísticas afectan a personas, corremos el riesgo de despersonalizar para que nos resulte todo menos doloroso. Tenemos que lograr que los mejores estudiantes de bachillerato conozcan todas las especialidades de la ingeniería, porque dado que nosotros enseñamos cómo se hacen las cosas, el por qué se hacen así y el por qué es necesario hacerlas, somos los profesionales que vamos a poder llevar a cabo, con la ayuda de toda la sociedad, los objetivos de desarrollo sostenible, y para eso necesitamos a nuestros mejores actores posibles. Querido Guy, querida Elisa, no puedo finalizar este discurso sin dedicar unas palabras en recuerdo de nuestra doctora honoris causa, la profesora Margarita Salas. La profesora Salas, de un campo que entiendo es muy próximo al vuestro, ha sido y es ejemplo de trabajo científico, de formación de nuevos investigadores e investigadoras y de transferencia de los resultados de esa investigación científica a la sociedad. Ha sido y es ejemplo de lucha contra las adversidades y contra los prejuicios que tenía la sociedad española respecto al papel de la mujer. Ha sido y es un ejemplo de cómo la investigación básica también produce retornos económicos, siendo titular de la patente que mayor rendimiento económico ha tenido en este país. La profesora Salas ha sido, además, maestra de toda una cohorte de nuevos científicos españoles. Descansa en paz, profesora Salas. Querido Guy, déjame que me dirija por un instante a tu familia, porque para nosotros es un honor y un orgullo que te acompañen en este acto, tanto tu mujer como tus hijos. Querida Betty, queridos Litze, Wouter y Dries, como rector de la Universidad Politécnica de Madrid es para mí un honor y un orgullo que vuestro marido y padre, respectivamente, haya aceptado incorporarse al claustro de nuestra universidad. La Universidad Politécnica de Madrid está orgullosa de vuestro padre y marido, pero sentid este homenaje también un poquito vuestro, porque como suelo comentar en las ceremonias de entrega de diplomas, para llegar a este punto, y él lo ha dicho también al final de su exposición, vosotros habéis tenido también mucho que ver en esto. Una parte de ese birrete, de esos guantes blancos y del libro de la ciencia, pensad, y él lo sabe y lo ha dicho, que también os la habéis ganado vosotros. Enhorabuena. Ya para finalizar llega el momento de los agradecimientos. Quiero agradecer a todo el personal de la universidad que ha hecho posible la celebración de este acto. Personal de protocolo, de servicios centrales, de audiovisuales, de comunicación, de las diferentes conserjerías, del gate de las escuelas. Quiero agradecer también, como siempre, a nuestro coro, el coro de la Universidad Politécnica de Madrid, que nos ha acompañado en este acto. Como les digo siempre, la música no es decorativa, es parte integral de los actos académicos. A todos ustedes, asistentes al acto, muchas gracias. Nos ponemos en pie para escuchar solemnemente el himno universitario, por favor. Muchas gracias. El acto ha terminado. Se levanta la sesión.